All right, today we're diving into a real-life agricultural detective story. We're going to be cracking the code of a mysterious and pretty devastating olive tree killer.
So let's set the scene. Just picture this: ancient, centuries-old olive groves, the kind of landscape that defines entire regions, suddenly just withering away, basically turning to dust. That's our crime scene, and the stakes, they couldn't be higher.
Okay, so what exactly are we dealing with here? In this first part, we're going to unmask this invisible threat, something that has been spreading silently but absolutely systematically.
Meet our main suspect, Xylella fastidiosa. And honestly, you can think of it as a microscopic clog from hell for plants. It gets inside a plant's vascular system, its veins, and it just creates this massive blockage. Water can't get through, nutrients can't get through, and the plant essentially just starves to death right from the inside.
So, like any good investigation, you got to look for the clues, right? The evidence left at the scene. And what scientists see are these telltale signs. The leaves start turning yellow, the tips look like they've been scorched by a flame, and entire branches just suddenly die off. But the real calling card, the most distinct clue, is this leopard pattern of dead spots. When you see that, you know who you're dealing with.
And here's the thing that is truly staggering. This isn't just about olive trees, not by a long shot. We are talking about over 600 different species of plants that are vulnerable to this thing. It just doesn't discriminate. It hits almonds, grapevines, cherries, even the oleander you might have in your backyard. This is a massive, widespread threat.
Okay, so once those clues are spotted out in the field, the real forensic work begins. The investigation moves from the grove into the lab. Let's take a look at how scientists actually follow that trail of evidence to get a positive ID on this killer. And the process is super methodical, almost like a police procedural, right? It all starts with that visual inspection we just talked about. If things look suspicious, they move on to what's called an ELISA test. That's a quick screening that looks for proteins linked to the bacteria. But for the final nail in the coffin, the absolute conviction, they use a PCR test. And that test looks for the bacteria's actual DNA. There's just no arguing with that.
So we've got our culprit. We know how to identify it. But there's a huge question still hanging out there. How does a microscopic bacterium travel so fast? How does it get from tree to tree, let alone cross entire regions? Well, it needs a getaway vehicle.
And here it is. Meet the accomplice in this whole crime, the spittlebug. This tiny little insect is the primary vector, the main getaway driver. And its method is so simple but so devastatingly effective. It just feeds on the sap of an infected plant, picks up the Xylella bacteria, and then it flies off to a perfectly healthy plant and injects its next victim with a fatal dose.
So the obvious question is, how do you fight back? And honestly, the reality here is really sobering. The first line of defense is just brutal containment. We're talking cutting down sick trees, trying to control the spittlebug population. But here is the most crucial point of all. Once a plant is infected, there is no cure. It's a goner. You simply cannot save it.
But this is where the story pivots. Because if you can't cure the disease, you have to change the entire game. So we're moving from a purely defensive containment strategy to going on the offense. And the new battlefield? It's genetics.
And it all started with this one powerful question. Instead of trying to kill the bacteria inside the plant, what if we could find a plant that already knows how to defend itself? What if the solution wasn't something we had to invent, but something that was already hidden out there in nature? And then they got their first huge break. Scientists started comparing the genes of all sorts of different plants, olives, almonds, you name it. And they found something absolutely remarkable. There was a set of 18 specific genes that kept popping up in the plants that were resistant. These genes were all tied to fighting stress and infection. It was like finding a genetic roadmap to survival.
Now, having a roadmap is one thing, but you still need a way to read it and use it. For that, scientists turned to a tool that is straight out of a forensics lab. They started using genetics as their primary forensic tool.
It's called sequence alignment, but honestly, the best way to think about it is DNA fingerprinting. It's the exact same idea. You take a little DNA sample from a sick plant leaf, and you compare its genetic sequence to a massive database of all the known Xylella culprits. It is literally like running a fingerprint from a crime scene against a police database.
And this is that aha moment. They run the sample from the olive leaf, and bam, a perfect 100% match against the reference DNA in the database. The proof is undeniable. The killer is identified, right down to its specific strain.
case closed.
And this technique is just a game changer for a few huge reasons. One, you get early detection. You can find the bacteria before the plant even starts to look sick. Two, you can identify the exact strain you're up against. And three, and this is the most important one, you can use it to find those rare, special plants that are successfully fighting back all on their own.
And all of this forensic work, all this genetic digging, leads us right here to the climax of the story. Because with these new tools, after years of searching, the heroes of this story finally started to show up.
So, let's meet these genetic heroes. We've got two Italian olive varieties, Licino and FS17, that were discovered to be naturally resistant. The Licino is amazing. It actually traps the bacteria inside its veins, it walls it off so it can't spread. And then, building on that success, scientists have even bred a new hybrid called Lechiana, specifically designed to carry that resistance forward.
And suddenly, the ultimate solution, it becomes clear. The long-term plan isn't about an endless war with chemicals and cutting down trees anymore. It's actually about hope. It's about replanting these devastated groves with these new, tough, resistant heroes that know how to fight for themselves.
So, we've cracked the code for this one killer, which is amazing, but it really makes you stop and think, doesn't it? With this kind of genetic technology, what other massive challenges, whether it's in agriculture or medicine or anywhere else, what other mysteries are just sitting there, waiting inside a DNA sequence for us to solve?